Welcome to the Kiromic BioPharma presentation today. My name is Tony Tontat. I am the CFO and COO of Kiromic BioPharma. Also on a call today, our team members, Dr. Maurizio Chiriva, who is a scientific founder and the CEO of the company. He's currently the associate professor at MD Anderson, and he'll be joining us on the call. On the call also is our Chief of Strategy, Mr. Gianluca Rotino, who is a specialist in patent prosecution, who will be joining us. And our Chief Medical Officer, Dr. Scott Dalbeck, will also be joining us on the call. Welcome to our call. Thank you for taking the time to learn about the Kiromic story. So instead of giving the full presentation, what I'm going to do is to give you the generalist modules. So I'll go through the modules in turn. They are the technical module lasting approximately 2-3 minutes. And then the financial module lasting approximately 2 minutes, and then a catalyst module lasting approximately 2 minutes. And after that, our full team will be on to give the Q&A the proper attention. So here we go. So I'm going to look at my notes on the side. Hang on. And if you are thinking about cancer, please know that there are different types of cancer. So the first is liquid cancer, which are cancer cells floating through our circulatory system. After that, there are cancer cells sitting on the very organs that we all have: liver, ovarian, prostate, lung, so on and so forth. Kiromic is solid tumors, cancers. Next, when you're treating cancers, there are also the small molecule version and the live cell therapy version, also known as CAR-T. You've heard about it for many years. In small molecule, these are cooked up in a laboratory and injected into the patient to kill cancer. In the live cell therapy, the CAR-T, these are T cells extracted from a patient or a donor given to a patient, also known as CAR-T, chimeric antigen receptor. Kiromic is CAR-T, chimeric antigen receptors. When it comes to CAR-T, there are also the autologous version, which is very much like a hair transplant or a skin transplant. It means that from the patient to the same. In autologous, T cells are extracted out of the patient, gene edited, expanded, and given back to the same patient. That's autologous. In allogenic, the T cells are extracted out of a donor, gene edited, expanded, and given to the patient. Kiromic is the allogenic CAR-T version. Within allogenic, there's also the one-to-one relationship in which it works like an organ transplant, whereby T cells are extracted from a donor, gene edited, and given to one single patient. The other version of allogenic is one-to-many, otherwise known as off-the-shelf. So in off-the-shelf, a single donor gives T cells to Kiromic. We gene edit that, we expand that, and that after the expansion, the same T cells donation is able to treat up to 500 patients. Okay? And so now that you know, Kiromic is solid tumors, allogenic CAR-T, off the shelf, that you need to know. Next, for treating cancers, scientists also need to differentiate the difference between cancer cells and healthy cells. Biomarkers, which are little protein fingers, expressions on the surface of cells. These are called biomarkers. So in the old time, biomarkers, the journey going through from discovery through validation takes approximately 3 years and up to $100 million. In the case of Kiromic, we have a proprietary artificial intelligence engine that can accomplish the same discovery, validation in a few hours. So that's it. That is a Kiromic technical module. Next, I'll give you the financial module of Kiromic. So on the 16th of October, 2020, Kiromic went public. We had sold 1.25 million shares at $12 a share, yielding $50 million gross and approximately $13.23 million net to Kiromic. And our monthly burn is approximately $650,000 to $750,000 monthly. That takes us our cash runway to go from October 2020 all the way out to June or July 2021. And so that said, in the absence of us having to manage and pay for severe adverse events on our trial, our cash runway is likely to be able to extend all the way to September, October of 2021. So that concludes my financial module for you. Next, we're going to cover the catalyst module. In our catalyst module, you know that we had on our S1 disclosed that we would be filing 2 IND with 2 biomarkers, CH-PD-1 and isomesothelin for ovarian cancer. We did that on the 17th of December 2020. Going into 1Q 2021, we are looking forward to announcing the first in-human dosing of our off-the-shelf allogenic CAR-T for ovarian cancer, and we'll be updating you on that. And into summer of 2021, we will be conducting our combo trial of CH-PD-1 and isomesothelin in ovarian cancer at the same time. So those two in the same trial. So that said, that concludes our technical, financial, and catalyst modules. We would like to open the call now to Q&A. We have our CEO and scientific founder. Please go ahead, Nathan.
Sure. Thank you, Tony, and thank you for taking the time to present today to the audience and for joining. So a few questions coming in so far. What are Gammadelta T cells and how are you using them? How are you using them in your CAR-T?
Okay, Dr. Chiriva, please go ahead. You're on mute, sir. Can somebody unmute Dr. Chiriva, please, if it's available on? Okay,
hello.
Please unmute it.
Good morning, everybody, and thank you for the questions. So the first question is how is being used the Gammadelta T cells in the context of CAR-T. Am I right, sir?
Yeah, well, we could start there. There's a follow-up to that, but we could start there.
Okay. Gammadelta is a subpopulation of a T cells with a unique capability of acting as an NK and also a T cells. We have a very unique next generation of manufacturing of these little fractions that is constituting our immune system, that is the gamma delta. The gamma delta, when being selected from healthy individual, can be you, me, anybody, we have a proprietary technology which allow us to select and expand. And then from that particular expansions, we can transduce what it means. We can engineer or introducing gene into the gamma delta, which has the expression of that particular target for which we are going after, specifically for solid tumor and in case specific for ovarian cancer. So the gamma delta has a very unique capability of able to kill almost twice, one with the arm of an NK of immunity, and the other through the engineered target that has been inserted into the cells.
Does that answer your question, Nathan?
Yeah, I think so. As far as, so if you take ovarian cancer as a solid tumor, and you're using CAR T Gammadelta T cells, how does that work to attack the tumor? How are you getting to the tumor site? Give an example of that in a real life setting as far as ovarian cancer goes.
Okay, we do have a cartoon that shows exactly the chart of how it's actually being given to the patients. So what we do, we have a manufacturing in-house in Kiromic's headquarters in Houston, a manufacturer GMP, where we generate our, exactly this slide. We engineer and expand our Gammadelta T cells, as you can see here in the cartoon on your left, actually on your right, my left, which then they are completely harmed, this being called, and put it into a little bag, really a small little bag that contains the cells engineered, our gamma delta, and then it's being given IV through the portal to the patients, simply in a real time. Normally, it's an outpatient kind of treatment. In other words, you don't need to be hospitalized. And talking with our sites, our clinical sites here in US, from MD Anderson's and other specialized clinical sites, they will basically do an outpatient, few hours, following the patients. And because it's such a safe treatment, because the gamma delta has the uniqueness of not generating GVHD or collateral, and therefore they do not have toxicities. So that can be extremely valuable, having an off-the-shelf, and these little bags that you can see are being stored and generated basically ahead in our facility, of which we can use it as needed and send it to the site, and they can basically just thaw the cells and give it to the patients.
So Nathan, can I help you reframe some of this information, which is fairly scientific? And I'm a generalist myself, so I'll give you this version, right? So it starts out with a concept that two cells must bind to each other using chemical signals. Does that make sense? So on the surface of a cancer cell, there's a receptor. There's a protein expression. On the surface of the CAR T cell, there are the receptors, and they bind to each other. That looks something like this, okay? The T cell normally don't have the knowledge to know that it needs to do that. That's what the gene editing does, right? Into the gene edit, Dr. Chiriva and the scientists at Kiromic are going to insert that information. So as soon as the CAR T is inserted, gene edited, it has that information, immediately it goes and extrude the receptors that matches up perfectly with the protein expression on the cancer cell. Once the two latch together, normally T cells kill cancer cell by releasing these things called perforins. Perforins come in and they're like little drill bits. They light the membrane of the cancer cell. The internal of the cancer cell spills out, macrophage clears that out, and that's how T cells kill cancer cells. And so that's it. Once we get the donor tissue, whole blood, directly from the patient, right? We go to the lab and we do the gene edit. With the gene edit, the T cells now have the knowledge to be able to, as soon as it approaches the cancer cell, it does something like this, right? It comes in, boom, receptors come out, latches on to the protein expression on the cancer cell, performs go between the T cell into the cancer cell, lyses the membrane, and breaks it down, right? But that's it.
How do you get localized to the tumor site?
That's a localization. That's a localization. Normally the T cell would just float around,
right,
not knowing what to do. But because of this gene edit, it knows to say, okay, the chemical is pulling in, 1, 2, 3. Receptors come out, boom, it latches on. That is a localization word I think you're looking for. How does it get to the tumor?
Yeah, I think Mr. Tontat exactly explains how the gamma delta, when injected, goes to the tumor. It's called actually homing. Normally, T cells in general, especially gamma delta or NK, they are basically traveling through our blood, and they're going through the system as or almost a soldier, as an immunosurveillance act, which will encounter the tumor that express the cancer, as was mentioned by Mr. Tonta, and that's exactly there where there is a connection. It's like any other mechanism of action of our immunity, but this is specifically engineered, of which the gamma delta, engineered by Kiromic, are seeking the specific target. It is definitely a very unique system which allow us to go to the tumor site. And that when we are injecting it, the tumor, through an IV, through a blood a portal. But you can also, in our clinical trials, we have also an injection that is site-specific, and that it could be valuable for ovarian cancer in the IP on the intrapleural area, or for mesothelioma on the pleural, on the upper body of the intestine, and that can be injected specifically there. Of course, we are working with specific center for the site-specific injections. We doing both type of sites. One is IV, just general injection, and the other is site-specific.
On ovarian, because that's a good example, are you looking for a certain type of expression in those particular patients in order for your technology to be able to get to the tumor site and be effective?
Yeah, this is a very good question. For ovarian and for the mesothelioma, we are looking at to express our target, our variant isoform of mesothelin, and that it's highly expressed in mesothelioma and ovarian cancers. These are the two indications. For the PD-1 expressions, we know already that the profile of the solid tumor, specifically for ovarian cancer and for mesothelioma, is pretty high. So we look for these two targets, highly expressed, and we treat the patients based on the expressions of the individual. That it's quite high expressed, so therefore we don't foresee any issue in the enrolling patients. Most of the patients will be expressing these targets.
Understood. As far as Gammadelta T cells, has anybody else used these cells in the past? How have they progressed, and what does that landscape look like?
So, Dr. Chiriva, I'll jump in very quickly, right? So you know, Nathan, when it comes to CAR T, right, the standard way for CAR T, and remember, CAR T was a concept back in November 2014 when it came out at American Society of Hematology, right? Right then you have Kymriah, Yescarta, which are now current drugs, right? Owned by Juno and Kite back then, correct? It seemed like a novel concept. People say, how is it possible that you can extract T cell away from the patient, gene edit that, and give it back to the patient, one single shot, and it cures cancer, right? That puzzle has been solved, correct? So since now we are in 2021, CAR T is now like a 6-year-old technology. It's no longer that concept, right? So now when they extract out T cells from the patient, gene edit that, they found out that many of the patients are so weak that the T cell that they donated to be gene edited makes a poor candidate. Does that make sense? Mm-hmm. And another issue came up in the old CAR T is that the old targets only allow CAR T to access hematologic cancers. So now we're moving on to Kiromic, right? So Kiromic says that old CAR T, weak T cell donation from the patient doesn't make a lot of sense. And the whole entire scientific community came to agreement on that as well. So they're saying, why don't we take T cells away, directly from a healthy donor and use those T cells, gene edited, and then give that to the patient. That makes more sense, right? Because now you have a healthy donor with a strong, robust T cell system. You gene edit that and give to the patient. It makes sense, right? But now you come out with 3 choices, right? So Nathan, they have choices between stem cell, NK, and Gammadelta T cell, right? So pulling them out, that's an easy part. Gene editing also seemed easy. The challenge is in the expansion, right? So it turns out that we tested stem cell, we tested NK, right? So stem cells are, they tend to take the signal directly from the cells in which they're next to, right? So to gene edit them, torque them into behaving a certain way, more difficult, right? NK cells, they are natural killer cells. They multiply very quickly, and because they are immortal, they don't have apoptosis, right? So they could turn out to be difficult to deal with because they become cancer cells themselves. In order to deal with an NK cell that has immortality, they have to radiate it. After you radiate it, it's not so potent anymore. So now those are challenges with these two, stem cell and NK cell. Now, Dr. Chiriva talked about gamma delta. It turns out that gamma delta are the most, or I shouldn't say not the most, but the easiest one to expand ex vivo after the gene editing and still have a very high yield. So that's why we use gamma delta. Does that make sense, Nathan? I hope the answer is not too long.
Sure. No, that makes sense. And can you talk about, with that being said, because this is somewhat autologous but allogenic at the same time, because you're taking it out of the patient. Can you talk about cytokine storm?
Yeah, so I'll jump in also as well, okay? So Nathan, we have to make sure that in Kiromic, we know that it is solid tumor, right? After that, we know it's allogenic, right? After that, we know it is off the shelf, right? And then after that, we know that, we deal with a microtumor environment, right? So those things have to be solidly locked in place. In Kiromic, we can never get confused between autologous and allogenic. Kiromic is 100% allogenic.
Why?
Right?
Because you're changing the cells? You're editing?
No, it's allogenic because it comes out from the donor.
Oh, it is donor. Okay.
Yeah, it is donor-based.
You're not getting it from the patient.
Yes. Because getting it from the patient, the T cells are too weak and fairly difficult to work with gene edit. And this, I tell you, the whole entire CAR T universe in research and also in the clinicians, they all know this now. Getting donor tissue directly from the patient themselves, getting T cell directly from the patient, very difficult to work with and fairly ineffective. That's why they have such a high refractory rate with the 2 CAR T that are approved, Yescarta and Kymriah.
Great. Makes sense. Some other questions. Where was the technology founded and by whom?
Dr. Chiriva?
The Diamond AI is being founded by Kiromic, by us, and the Gammadelta manufacturing, expansion and gene editing is being, expansion and the fraction edit is being founded by Kiromic.
Can you talk about the institutions where you're going to be running these studies, or where you're setting these, where you're going to be able to utilize your CAR Ts?
Yes. We are in the process of finalizing agreements with MD Anderson Cancer Center and with also a specific site that is specialized in phase 1 in San Antonio. They're specialized in phase 1 in clinical trials. And also we are in negotiation with the Texas Oncology, that is a nationwide structure, so which allow us to have multiple PIs around the country in the US. So we will have a multi-center study which will be primarily leading by MD Anderson Cancer Centers. We are negotiating right now with Sloan Kettering in New York. So they are prestigious sites and there will be a multiple of them, which will allow us to complete our first phase very quickly to enroll our patients in a swiftly mode, and therefore allow us to get to the process on the phase 1 and phase 2 quickly as we negotiate in these multiple sites. Also, we are negotiating at the moment with prestigious sites in Europe, which will allow us to expand the cohort of patients and populations. So we've been fairly lucky at the moment. So with the prestigious centers such as MD Anderson, Sloan Ketterings, again, we are negotiating right now. We are finalizing agreements. Of course, it takes time, but we've been working for the last year on this, and therefore we feel very comfortable that soon we will have this site up and running, and therefore starting with our first dosing.
Yeah. So Nathan, please think about the following, right? So when it comes to getting these sites, it's not different than any kind of persuasion that you have to do in sales, in investment banking. You imagine that you get one big investor on, right? It's very easy to go and say that, RA Capital Partners have signed on to be a lead investor in the next round. So guess what? You get a whole bunch of follow-on. So that said, the lead guy, they never give, the best valuation. So exactly the same as if we're negotiating with MD Anderson, they would strip everything out to the bone, right? I mean, that's a big organization. They're very smart. And so once we sign on to the big ones, all the other little ones are going to fall in place, right? So that said, it's likely that we're going to have a press release that would speak to the names of the sites that we have finally selected toward the first in human dosing. Does that make sense, sir?
Sure. Can you talk about the 2 phase 1s that you filed INDs for? Since you filed those in December, likely you should be getting a response on those shortly. And as far as ovarian solid tumors, are you going right into patients, and will you get some efficacy data in your phase 1 studies?
Yeah, so first we'll break the question into 2 parts, right? So first you want to know about the first 2 phase 1, correct? And then after that you want to know when the data is going to be available for those, right?
Yeah.
Are those the questions? So we get it right?
Yes.
Yeah, Dr. Chiriva, will you speak to the return of the FDA on the 2 IND that we had filed?
Yeah, we had also already a preliminary, since the submission of the first, and we had been in basically weekly communication with FDA, which they required, some adjustments as being requested. So, as you know, even with the pandemic, the FDA has the rule to address with a letter and then details in the 30 days, and then 30 days after that with the details. Normally, the FDA doesn't release, it's very difficult, they release a green light in at the first round. So we are expecting by the end of this month an official letter that is already being communicated to us via telephone and via email, of which they discuss it, what they are looking for. So we will see the details. We already know what they will ask to address few questions. Is it just normal procedures? Which I think will take us another 30 days. That is perfect in line to our forecast on our business model, which will take us on the end of March for the first dosing. So we do have, even again with the pandemic, I think the FDA is doing a great job to address any concerns, like in any phase 1. This is a novel study. So the process is in 30 days that is passed already addressed to us. They will send a detailed letter, so which they will address they will have some questions, and we already talked to them, and we already adjust some of them, and as soon as they will receive the letter, we will reply. We're already in the process of replying some of these questions on details of which we are already in process. And we are planning to resubmit based on their time, their reply, and this will be the functional process, so which we feel that we will have enough time based on our timeline to address
Nathan,
any comments.
I think that it's fair to count 60 days from the 17th of December.
Correct. Thank you.
And the readout of the first, Maurizio, would you like to speak to the timeline for the readout of the data once the dosing begins? What is the first available read, sir?
Yeah, after the first dosing, we will look at 30 days, of which, if you can see in our clinical program, so will allow us to go through the first 30 to 60 days after the first injections. We have a different injections, a 3x3 study design. What it means that we test 3 doses with 3 patients. So let's say the first level of dose is an example. Let's say we gave X amount of cells and the patient is responding, not just is safe, because this is the first thing we're going to look at it in the next 30 days of the first dosing, but also if there is a efficacy. Efficacy means that there is a response of specifically reducing the tumor burden. In other words, is really killing the tumors and reducing the tumors significantly. So we have 2 readouts: safety, there is no toxicity in the next 30 days of the first dosing, and then we are looking also in the next 30, of course there is a follow-up of the patients, but the first 2 readouts are safety, no cytokine syndromes, no any allergies, none of it, or just a minimum of reaction like a simple epidemic allergies of alcohol reactions, but that is standard, like having a flu shot. And then most of it, we are also looking for tumor burden reduction, means the efficacy of the treatment. In other words, we are looking at in the next, after the first injection, the 30, 45 days, really to check with an imaging, an MRI or a PET scan, the reduction of the tumor, specifically in this case, ovarian cancer. So
yes,
these are the 2 readouts we are looking at.
So Nathan, to make sure that we recap this, right? So the 2 things that we're looking for would be safety, the absence of cytokine, and the absence of allergic reactions, and any other, severe adverse events. From what you have seen in Kymriah and Yescarta trials, this has been very, very low, because this is a biologic. It goes from a human to another human. And then after that, for efficacy, we'll be looking for specific tumor reduction as measured by, the mass of the tumor or the diameter of the tumor before and after measurements.
We have 5 minutes left, so we have time for a few more questions. How many patients in the ovarian trial will there be?
Dr. Chiriva?
So we are testing right now for the first phase up to 18 patients, a minimum 12, with a minimum of safety of 3 patients. What it means is a 3x3. It means if there is no side effects, you can test up to 3 patients with the same dose. And if you have no toxicity and efficacy, you can technically go back to the FDA and ask to go to the second stage of the phase 2. Let's say that we have no efficacy on the first dosing. We have had 2 dose, higher dose, to test on a different patient. So it will be another 3 patients and another 3 patients. So at least, at the good scenario, 3 patients. At the complete of the pilot to the phase 1 will be to 12 patients, and then we keeping going enrolling patients because it's called Siemens-less phase 1, which the FDA will work with us on monitoring our patients, and which allow us to constantly enroll patients to achieve the statistical numbers to be able to approve in the course of the phase 1, and then seamless phase 1 become phase 2 with a large number of patients, up to 40 patients.
Yeah. So Nathan, to frame that question, that answer a little bit more, think about the following, right? So you have like different cohorts. This first cohort go through a few, 3 patients, they get a lower dose, just to make sure that safety is okay. Does that make sense? Next
Yep.
one, the next 3 patients, they get this dose, correct? And then the next 3 patients, assuming that safety profile maintain a similar trajectory, right? And then the last group of patients come through, get the highest dose. And it's not like a small molecule trial in which all patients get exactly the same dose, right? Here, if we go into phase 2, we would keep that the highest dose and just add more patients to it. So it is a very short pathway versus like the old way of doing small molecules trials, in which at the end of every trial, they shut the whole entire thing down, read it, and then restart a brand new trial. And you have that 6-month gap in between end of phase 1 to the beginning of phase 2. Right? So given the amount of time that we have left, Nathan, even if you haven't asked this question, I would like to go over very quickly the comparables of Kiromic so that it sinks in what makes sense to people when they walk away, right? So Kiromic, you already know, is a combination of AI technology that fixes the biomarker targeting, right? And it's also the the CAR T component of it that uses the same biomarker, but now a biologic, a cell from a human to another human to kill cancer, right? There's no company out there doing that, and I'll explain to you why. On the left side, at the lower left, you have Lantern Pharma. They have an excellent AI engine. You see they just raised $60 million by Think Equity, and they take their AI engine, they comb through a whole bunch of failed clinical trials, fish out the one they believe they can fix, and take that into the clinic, right? Next, it is BioXcel. They take their AI engine, comb through thousands and thousands of 505(b)(2) drugs that had gone off patent, fish out one which they can find a new indication for, and run a human trial on that. Excellent. They are valued at $1 billion. And then you have Black Diamond Therapeutics and Schrodinger. Both of them have AI engines, and they use small molecule to drug those targets, right? So that's it. On the right side, you guys already know the CAR T space. Everyone is valued in the multi-billions of dollars. Correct. So Kiromic today, standing at approximately $9, presents an excellent opportunity for investors. And I thank you all for taking the time to learn the Kiromic story.
Tony, thank you, and thank you for presenting today. Very exciting story. The questions I think could go on here because there's so many things you guys have to showcase with a new technology like this. So thank you, and we look forward to hearing more.
Thank you, Nate. Thank you, Noble, for hosting.